Chapter 21 deals with viruses for the most part, but it also mentions prions and viroids. So we'll talk about all three, but we'll spend most of the time on viruses. We're going to kind of describe what they are and their structure, go over how they reproduce, and talk a little bit about viruses and viral infections. Okay, viruses are non-living microorganisms. Some people don't consider them alive, but for us, we will consider them kind of on the edge. They are acellular. They have no cells. They have no energy metabolism. They don't make ATP or use ATP, or they're not heterotrophs or autotrophs. They don't move independently. They don't transport materials, and they can't reproduce without cells. They're really intracellular parasites that are chemical in nature. They must be in cells, so they're obligatory. They do have a genome like an organism, but unlike organisms, it could be DNA or RNA. They never contain both, and it can be in different forms than other organisms have. Other than that, they have a protein coat. That could be it. Some have a lipid envelope surrounding them that they get from the host cell. Some viruses, most viruses, will have protein spikes that extend from the coat or the envelope. Most viruses only infect one type of cell, and their host range is determined by specific host attachment sites on those spikes usually. Viruses are chemical in nature. They can be crystallized, and they are really small. We measure viruses in nanometers, so they can be dozens or hundreds of times smaller than cells. Okay, here's a picture of a virus infecting a cell. That's a prokaryotic cell there on the left there, and the virus is pointed out. So it's much, much smaller than a prokaryotic cell, which is usually only 2 micrometers across. So much, much smaller. There's a eukaryotic cell on the right with virus particles kind of coming out of that cell. Very, very small. That's a picture from your book. I like this one better. This picture comes from a different textbook, and here it shows a human red blood cells, one of our smaller cells, which is 10 micrometers in diameter. And then it shows a bacterial cell, which is 3 micrometers long and 1 across. And then it shows some viruses. Common viruses like adenoviruses or rabies viruses or vaccinia. Vaccinia is actually a big virus. They're much, much smaller than a cell. Okay. So let's look at the structure of a virus more closely. Inside a virus, there is nucleic acid. Okay, that could be DNA or RNA. It's then surrounded by a protein coat called a capsid. The capsid is made out of subunits called a capsomere. Okay, there may be an envelope, there may be spikes. In this virus you see here, there aren't any. Okay, it's just a polyhedral virus, which means a multi-sided figure, often 12 or 20 sides, and then the DNA inside. There could be some enzymes, some protein enzymes in there. There could be some other molecules that wrap around the protein, but that's usually it. Here's another kind of picture of a virus. This is HIV. This one has an envelope. You see the capsid there, the DNA inside, and some enzymes, reverse transcriptase, one of them being in there. But here I'm really talking about the genome, okay, the DNA or RNA. In viruses, it can be single-stranded. Okay, it could also be double-stranded. It could be one or the other, never both. The strands could be in several segments, or it could be just one segment. It could be circular, or it could be linear. There's a lot of diversity in viral DNA, and it makes it kind of hard to understand them. HIV has an envelope. It comes from the host cell. And sticking in the envelope are some glycoproteins that are needed to penetrate and enter other host cells, and those are viral proteins that are sticking out there. So HIV looks like that. That's another complex appearance of viruses. Okay, viruses can also be linear, or I shouldn't say linear, helical. So it's got a capsid that's where the proteins wrap around the nucleic acid and you produce a worm-like structure or a helical structure. Sometimes these are small, sometimes these are large. That's Ebola virus there. Okay, there's another polyhedral virus. Okay, you know, and it gives you a size bar there. These are all pictures with electron microscopes or diagrams. And here's another envelope virus, influenza virus. Okay, and there's herpes virus. All right, so these are really small, 80 nanometers. Some viruses, particularly viruses that infect bacterial cells, can have a very complex structure with a capsid head, a sheath, and tail-like structure with a base plate that is actually going to inject the DNA or RNA into the bacteria. These are often called phages because they are phage is something that eats them, and they were first seen because they eat into the virus or the bacterial growing on a plate. Okay. So again, virus genome. Viruses have very simple structure protein-wise, but the genomes can be complex. DNA, RNA, single-stranded, double-stranded, linear, circular, segmented, or non-segmented. Okay. I wouldn't expect you to know this Baltimore classification, but they classify viruses by their genome. Some places will classify them by their shapes. The genome classifications are becoming more and more popular because this also indicates material about their life cycle, how they reproduce. So here you have 7 classifications of virus based on DNA, RNA, single-stranded, double-stranded, then what happens to them. In general, when viruses replicate, there are many variations depending on the type of genome and what enzymes the virus possesses. Commonly, viruses must have several stages. Attachment, where they clamp onto a host cell or are attracted to a molecule on a host cell surface. Entry, where the genome enters into the cell. Then replication, where the host cell enzymes copy the virus genome and proteins. And then these things assemble and are released. Okay, usually viruses break open virally infected cells and are released from them. And so these cycles commonly look like this. On the top, there is the lytic cycle, where the genome enters the cell and gets the host cell to copy its proteins and its nucleic acid, okay, which could be DNA or could be RNA. Then the new virus particles assemble and cause the cell to rupture. This is called the lytic phase because the cycle involves automatic lysis of the cell. So infected cells are killed. Then the other type of cycle is the lysogenic cycle. And again, there's attachment and entry of the genome. But in this case, the genome is going to become incorporated into the host cell chromosome. Okay, in the case of bacteria, this is called a prophage. Okay, it's still virus DNA, but it's in the host chromosome. But if this is in a eukaryotic cell, we might call it a provirus. Okay, so whenever then the host cell reproduces, the virus DNA is copied, and any new cells that are made have the virus DNA in it. Okay, under other conditions, then the virus can then get out of the host chromosome and it can enter the lytic phase where many viruses are copied and the virus proteins are copied and new viruses assembled. Okay, and so you have these two phases, the lytic phase where the host cell always dies, and the lysogenic phase, where the prophage or provirus is incorporated into the DNA, and you can have this special life cycle. Okay. So don't worry too much about the different types of cycles that we have, but if you could remember the lytic and lysogenic, and then you don't need the Baltimore classification. Okay, here let's go over this with a human cell and an important infection in humans. That's human immunodeficiency virus, HIV. And in here, we know the molecules, HIV virus, that attach to the host cell. And then the host cell is engulfed because the membranes of the virus envelope and the host cell fuse. The virus then releases its RNA into the cell, and then a viral enzyme, reverse transcriptase, makes DNA from viral RNA. And that viral DNA can wind up into the host DNA, and so it can become a latent infection. Okay, it can also have the host DNA make RNAs from that, and those RNAs are then can be made into viral proteins, and the RNAs can serve as new host genomes. Okay, and there's an enzyme, again, another enzyme, integrase, that helps the virus integrate itself into the host chromosome. So the virus has several enzymes, and even another enzyme that helps the viral proteins be separated. Okay, and so the virus has three enzymes: reverse transcriptase, integrase, and protease. And drug treatment then will work on affecting those enzymes. So human immunodeficiency virus, two single-stranded RNA molecules produce DNA, okay, and that's done with reverse transcriptase. Okay, the virus also has enzymes integrase that help it integrate into the host genome, and protease, which cut the proteins into the right size. And like other viruses that have this integrase enzyme and it can be incorporated into the host genome, sometimes these viruses that do this cause cancer. Okay, and so they're referred to as oncogenic viruses. And so these activated, when these genes are put into the host genome, they can cause cells to transform into cancer cells. Okay, and so this happens with viruses that form a prophase or prophage or provirus. Okay, and many of these infections that also integrate into host chromosomes can produce long asymptomatic phases called latent phase, where there's really no symptoms. Herpes viruses are notorious for doing this, and because they're integrated into the host chromosome, they're hard for the immune system to find, and so these infections may last for a long, long time. They may become persistent or latent infections. Okay, so then let's go to prions. These are protein infectious particles. They're even smaller than viruses. These proteins contain no nucleic acid. They have no genome. And the only known prions are those that cause fatal neurodegenerative diseases. These include mad cow disease, which is called bovine spongiform encephalopathy, Creutzfeldt-Jakob or Kuru in humans that are spread through cannibalism, and then scrapie, which is in sheep. We also see chronic wasting in deer. And what happens with these is this is a normal protein that's found in the brains of mammals called PRP protein is converted into an abnormal form by the prion particle. And as these things accumulate, they damage brain tissue. And so this is a fatal neurodegenerative disease. Very rare in people, but a big problem in animals. In England, we had a big outbreak among farm animals, and right now we're in the midst of a big outbreak among white-tailed deer. Another form of virus-like particles are viroids. These are small circles of RNA that we are only known to infect plants. They're really small. They never have a capsid or are outside with any proteins attached to them. But they do cause crop failures that cause millions of dollars in losses in potatoes, I think squash, a couple other uh, domestic plants. So, in summary, viruses are small, non living, and acellular. They have a genome and a protein coat. They use host cells to copy their genome. Viruses' life cycles vary, and they cause human and animal diseases, which we'll talk about in our Zoom meeting.